This is an experiment about the digestion of lambda DNA with various endonucleases. To start the experiment, the gel must first be made. Prepare 600 milliliters of 1x TAE buffer by mixing 550 milliliters of deionized water with 12 milliliters of 50x TAE stock. Add enough deionized water to fill it up to the 600 milliliter volume. Measure out 100 milliliters of 1x TAE buffer and 0.8 grams of agarose powder. Pour the agarose powder into the TAE buffer. Microwave this on high heat until the agarose powder dissolves. This may take a few tries, so be sure to stop and stir the solution in between. Keep on repeating this process until the solution becomes clear. Once the agarose solution has cooled to the point where it can be touched, add 6 microliters of acidium bromide. Be sure to use gloves. A clean pipette tip can be used to pop any bubbles that may occur during the pouring process. Take the gel out of the casting tray once the gel has set and become. Now the DNA will be digested. Label 6 microcentrifuge tubes as shown below. Prepare the methylation tube first. Add in DNA, water, buffer, and EcoR1 methylase into tube number 6. The EcoR1 methylase will prevent the EcoR1 restriction enzyme from cutting the DNA later. Mix all 4 of these ingredients together, centrifuge to bring the ingredients down to the bottom of the tube, and incubate the tube for 50 minutes at 37 degrees Celsius. After incubating, add 1 microliter of EcoR1 restriction enzyme. Continue incubating for another hour. Prepare the other 5 tubes by adding 1 microliter of lambda DNA and appropriate amounts of 10x buffer, enzyme, and water to each tube. Follow this table to see how much 10x buffer, enzyme, and water should be added to each tube. So be sure to use clean tips every single time in order to prevent cross-contamination. Mix and centrifuge all 5 tubes. Incubate the 6 tubes at 37 degrees Celsius for 1 hour. After incubating the tubes, the cut lambda DNA will be analyzed using gel electrophoresis. First, add loading dye to the samples. Prepare the gel electrophoresis tray by pouring the remaining TAE buffer over the prepared gel. Be sure all the wells have been thoroughly covered. Load the ladder into the first well of the gel. This particular ladder that will be used is lambda DNA that has been already digested with HIN3 enzyme. Load the remaining samples in the order that they were numbered in. Be sure that the tip of the pipette does not enter into the actual well of the gel, otherwise the integrity of the well will be compromised. After loading the samples, run the gel at 100 volts for 1 hour. Remember to run to the red electrode. An arrow has been drawn on the picture to show that the wells are on the opposite end of the red electrode, so that the samples will run to the red electrode. After the hour has finished, take out the gel and image it using a UV light. Here is a close-up of the gel from this experiment. This lambda ladder has 6 bands, including 2 very faint bands at 2.3 kilobase pairs and 2 kilobase pairs, right here. Number 4, the control, looks as it should because it was not digested by any enzymes. This leads to a big band of thick DNA. Number 6, the methylase experiment, also worked because the DNA was not cut. This indicates that the EcoR1 methylase protected the EcoR1 cut site by properly attaching a methyl group. Number 3 is lambda DNA cut by HIN3, so it should look exactly like the latter, which it does. Number 5 should look as if the band patterns from BAMH1 and HIN3 have overlapped each other, since number 5 was cut by both enzymes. Overall, this experiment was a success and the results were as expected.